Okay, hi Year 13s. Unfortunately, earlier today when we had our Teams meeting, I did not press the record button, so now I'm recording a little video for you about what we did, just in case you couldn't make it to school. Now, number one, some of the general feedback that I had to give to you guys from all the drafts that I looked at, you need to please remember that I want a sugar homeostasis diagram, not just a generalized homeostasis diagram. It has to be on how the blood glucose levels are maintained. Okay? And then, in talking about that, that is actually on your tick list, on the grading sheet. So please make sure that you have that document open and that you are satisfying all the requirements for all the ticks in there. Okay, so what I did with the class was we actually went through and looked at a flow diagram of the whole system. So I do think this is important so that you can see the biochemistry and you can see how it then plays out and the kind of level that I want you to get into. So here we go. So you start off by eating. When you eat, obviously your food is digested and it gets into your small intestine. In the small intestine, it is absorbed. Now, it is absorbed in the form of molecules. You don't have little pieces of Big Mac floating around in your blood. It is broken down to the molecular level, and then in the small intestine specifically is where the glucose is then absorbed. It goes into the portal vein, and if the glucose has now entered the portal vein, you can see that the concentration of your glucose in the portal vein has just gone up. Makes sense, it's just been absorbed. However, the concentration within the beta cells that are in the islets of Langerhans in your pancreas, they are lower now than what the glucose concentration is in the portal vein. That means that you now have a concentration gradient that has happened. Okay, it is no longer in equilibrium. So as the concentration gradient is formed, because the blood glucose level is higher than the cytoplasm level in the beta cells, your glucose will flow down the concentration gradient and into the beta cells. And the way that they do that is through facilitated diffusion of the GLUT2 plasma membrane protein. Okay, so that channel is always there. However, when we are in equilibrium, glucose will not flow in or out, or actually the in and outflow of the glucose is equal. However, when there's more glucose in the portal vein, we have a concentration gradient that develops and it flows into the beta cells through the GLUT2. When it is inside the GLUT2, it binds to a receptor, and that receptor then stimulates the vesicle that contains the insulin to move towards the membrane. And as it moves towards the membrane, it actually merges with the membrane. And you need to remember that a vesicle is basically made up of a piece of membrane. So it's a little bubble made of membrane containing insulin that was made earlier so that you can have a rapid release of insulin. So as it merges with the membrane, we call that exocytosis. And that is how it then is released into the bloodstream. So the vesicle is stimulated to move to the membrane, merges with the membrane, and then it released through exocytosis. Okay, so that is the first part, how an increase blood glucose level is detected by your beta cells in the pancreas. Then the insulin is in the blood. Now, your insulin concentration obviously in the blood has gone up, but the blood glucose concentration is still the same. It is still elevated. So the insulin flows around the body, and now it is looking for an insulin receptor. And we find these on various different cells. However, the first one that I'll discuss is when it hits the insulin receptor on your liver cells. Okay, because liver cells and brain cells also have GLUT2, the transport protein or channel protein. So the insulin binds to the insulin receptor on the extracellular part, and then intracellularly it causes phosphorylation and a phosphorylation reaction. And that phosphorylation reaction actually activates the enzyme, and that enzyme's name is glycogen synthase. So, glycogen synthase. I am making glycogen. That is its name. And that is going to make glycogen. And we call that process glycogenesis. So because of the insulin, we have now entered glycogenesis. And remember, glycogenesis means the beginning of glycogen. Okay, so now if I'm using the glucose to build glycogen molecules, what happens to my glucose concentration in the cytoplasm of this liver cell? Yes, it drops. Your concentration drops, and because the concentration has now dropped because it's being converted into glycogen, this changes the concentration gradient, and suddenly the concentration of glucose in the blood is now again higher than the concentration of glucose in the cell. So now the glucose flows into the liver cell, and then glycogen synthase comes and takes the glucose and builds more glycogen, and the concentration drops again, and that is how the concentration gradient helps the glucose to move into the cell. In the brain, however, it also has GLUT2, and the GLUT2 is always open because the brain is exceptionally energy hungry. It takes 20%, 1/5 of everything that you put into your mouth. All that energy goes to your brain. So the glucose in the brain, the way that your concentration inside the cells are going to decrease is simply by the brain doing respiration. That then creates the concentration gradient, and when there's a concentration gradient, the glucose will flow into the brain cells. When we get to muscles and fat, that is slightly different. Now, the fancy word for fat is adipose tissue. So if you want to not use fat cells, you talk about adipose tissue. Now, they also have an insulin receptor. However, they don't have GLUT2 that sits in their membrane already. So it needs to be placed in the membrane. So insulin hits the insulin receptor. It then causes a phosphorylation reaction, which is called the transduction cascade. That transduction cascade actually now activates the vesicle that contains the GLUT4 channel protein. Remember, it's a vesicle that has all of these in its membrane, in the vesicle membrane. And then that flows or is moved towards the cell membrane. It merges with the cell membrane, and now we have GLUT4 channel proteins available for glucose to freely float through. However, when we think about the concentration gradient, before there was GLUT4 in the membrane of the cell, the inside of the cell will have a lower concentration of glucose than the outside of the cell. But now we have a lot of channels, a lot of GLUT4s, so where's the glucose going to go? From the high concentration in the blood into the low concentration of the cell until it reaches equilibrium. And when it's inside, basically if it's a fat cell, now fat can be made by joining the carbohydrates to 3 glycerides to make a triglyceride. Okay, and by making fat, you decrease the concentration. If the concentration of the glucose goes down, we create another concentration gradient, and the concentration gradient causes the inflow of more glucose into the fat cells. Muscles, on the other hand, they are very active. Well, if you use them. So they use the glucose in respiration, and as they use it, it decreases the concentration of the glucose intracellularly, and more glucose will flow in because of the concentration gradient. Can you see how I'm going back to the concentration gradient all the time? Because that is what the biochemistry is about. Okay. So, and also muscles can actually, when you exercise, you stimulate the GLUT4 to go to the surface, because now your muscles are working really hard. Lots of respiration is needed because lots of ATP is used, and consequently glucose needs to come in so that you can make the ATP. Alright, now I want you to remember that when we talk about hypoglycemia is actually below a certain level, and that level is 3.3 millimoles per liter. That doesn't happen often. That's not a natural thing to happen. Hypoglycemia does not mean I'm hungry, okay? It means you've been fasting for a long time, or there's something wrong with you. Alright, whereas hyperglycemia is when you've just ingested, let's say you're working through the night to get your internal done, and you're drinking 3 or 4 Red Bulls or something stupid like that, and then you have this tremendous spike So that is called the interruption, okay? It's called the disruption. It's not just the normal hemostatic control, because normal hemostatic control is small ups and downs, which is maintained between the, breakfast and lunch and dinner, and you know what I mean. Okay, so don't confuse the two. Hypo and hyper is big changes, and that is the disruption. Okay, but now in a normal case, you have just eaten. Now all the glucose is being taken out of the blood. It's being used and so forth. And because the glucose is now out of the blood, in your alpha cells back in the pancreas, it now notices that, hey, the blood suddenly has a lower concentration of glucose than what I have inside my cell. And the glucose wants to flow out through the glut to channel protein. Consequently, again, there's a concentration gradient difference and the glucose flows out. What happens then is the alpha cell says, oh my goodness, release the glucagon, because we need to slowly lift, or just tiny adjustments, lifting the glucose so that everything can still keep on running. So the glucagon is released by the alpha cells, it flows through the body, and on the liver it hits the glucagon receptor. That glucagon receptor then on the intracellular side, it causes a phosphorylation reaction, which then activates the enzyme, and the enzyme's name is glycogen phosphorylase. Okay? And glycogen phosphorylase drives a process called glycogenolysis. Remember, lysis means to break things. So we're breaking glycogen down into its building blocks, and remember what the building blocks were? They were glucose. So if I'm breaking down the glucose inside the cell, my concentration of glucose inside the cell will go up. Suddenly I have more glucose inside my cell than outside in the blood, so there's a concentration gradient. And what happens? The glucose flows out and can be used by the brain or muscles and so forth. Okay, I really hope that this helps you. Please remember to work with the tick sheet. And yeah, good luck, and I will see you on Tuesday. And hopefully by then you've got a good document to hand in at no later than 3:45. Okay. Thank you guys. Keep well. Be safe. Stay healthy. Bye.